let me start with something that has nothing to do with neuroscience, but it is very important. It's from Adam Smith. I think everybody knows Adam Smith for another book, the masterpiece that he wrote, The Wealth of Nations, The Origin of the Wealth of Nations. But before that, he wrote also The Theory of Moral Sentiments, which is a wonderful book. And here he wrote, how selfish soever men may be supposed, there are evidently some principle in his nature which interests him in the fortune of others, and render their happiness necessary to him, though he derives nothing from it, except the pleasure of seeing it. Here there is one part which is extremely important for a neuroscientist. If you read here, there are some principles in his nature which interest him in the fortune of others. So that's somehow implied that inside ourselves there is some mechanism, some principle, which render the fortune, as he says, or what you are doing important. So we are not isolated person living in an isolated world, but we are continuously connected with other people. Well, what we found some years ago, that indeed, of course, that's mere introspection. He was a genius, it was a great introspective work, but it's mere introspective work. What we found, there is indeed a mechanism which somehow link one person to another, one individual to another. We started our work in monkey, and we recorded, at the moment it may sound a bit strange, but you will see it makes a logic, it may have a logic. We recorded from the motor system, because motor system is something more complicated than people think. So we recorded from the motor system, and in particular from an area which is called F5. That's something that we changed from the traditional way. We call it the motor areas, frontal 1, frontal 2, frontal 3, just to stress that they are not merely area which produce movement. Anyway, we recorded from area 5, and the technique that we adopted is the recording of single neurons. So there is a technique which is common in neuroscience. You insert a kind of small needles inside the brain, and you can record from one neuron at a time. One neuron at a time is fundamental because each neuron has his own story to tell us. So when you have one neuron, when you record from one neuron, you test and you try to see what this neuron says. It's very easy because the code is just a modulation frequency. So if I put a needle inside my arm and I am able to record from a single fiber, and then I touch myself, I will hear tack, 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 tack. So what changes is only the number of tack, tack. If I put in my visual cortex, it will be the same, but the stimuli appropriate will be light. If I put in my motor cortex, it will be again tack, 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 but in this case it will be the movement of my head. So the code is called an action potential, the generation of action potentials. And these action potentials are the way in which the brain speaks, talks. And you understand that it's important because in this way you generalize. You have stimuli, you have motor activity, you have thinking, but everything is unified by the fact that the code in which the brain speaks. Well, now I will show you immediately what it means to record single neurons, so you will understand what I am talking about. Here it's a monkey. I hope you could see sufficiently well. Sorry. That's a grasping neuron. Well, so you heard now this tack, tack, tack. And what we do when we study the nervous system, we try to correlate this sound, this noise, with the activity of the monkey. In this case, it was obvious that when the monkey tried to grab something, there was a discharge. This is a very intellectual monkey. She's also able to use a tool. Maybe somebody of you noticed that when the monkey was using the tool, but there was no object, there was no discharge. When there was this small piece of an apple, this white thing, there was discharge. That's because this neuron code not the movement, but the goal of a motor act. I will come back in a moment to this point. So one of the properties of this area 5, of this premotor cortex, that it is that this area does not cause simple movement, but what is called motor act. What's the difference? Movement is something that you can elicit, for example, by electrical stimulation or magnetic stimulation of the brain. It will be something like that, twitches, displacement of an articulation. And when we use our head, we use a lot of movement. I use the same movement when I am scratching or if I am grasping a glass of water. It's always the same movement. However, in this case, for example, is this neuron, the only thing which is able to trigger the neuron, it's a motor act, the goal of action. You see why I say the goal? Because here it's in A, the monkey is grasping with the mouth, in B, with the right hand, in C, with the left hand. So it's impossible to explain the behavior of this neuron in terms of movement. It should be something higher. And this something higher is motor act. So the goal. That's a concept which is fundamental for understanding the rest of my lecture. So neurons in several of these motor areas do not code simply movement, but code motor act, the goal of a series of movement. Well, our approach was a bit different from the classical approach to the motor system, because we tried, I call it ethological, I don't like the word maybe too much, but anyway, it's ethological approach. So we tried to discover which is really the stimuli which are appropriate for the motor cortex. The classical point of view say, well, the motor system is for production of movement. We have to study the kinematics or the dynamics or some mathematical function of the motor system. Our view was different. We decided to see what happens in the real life with this neuron. In the real life, we discovered at first, I already told you, that they don't call movement motor act. The second thing, which is explained here, that there are many of these neurons which discharge when you present visual stimuli in a specific way. So if you have a neuron which fires for precision grip, if you present a raising, it could be a discharge, although the monkey is completely still. If you have a neuron which fires for whole hand branch, like for an apple, and you present an apple even 1 meter far from the monkey, there is discharge. So what it means? It means that this system transforms visual stimuli in the appropriate potential motor act. The monkey is not moving. It's a potentiality. So when I see object, my motor system already, it's ready to perform a series of motor acts, which is a big advantage. So you don't have to think how I have to move my hand. It's automatic transformation of object into a type of movement. Well, up to now, however, there was something which it was said that this was a breakthrough, our discovery. It was not a breakthrough, it was very interesting stuff, but somehow physiologists predicted that it should be this, because our behavior is so fluid. What we have not predicted, and it was a surprise for everybody, was that there were some neurons, which are now called mirror neurons, which fire both. One, the monkey was grasping, as in B, or the person was grasping. So the monkey was looking at the experimenter, and the same neuron, I'm speaking about the same neuron, was activated. Again, I will show you a movie which is the best demonstration of what I mean. Now, again, what you have to do is correlate the activity with the noise. At the beginning, you will see a grasping neuron, similar to the one you already saw, and then you will see the phenomenon. We managed to do it. So what you see here, now is the monkey grasping the object. That's the mirror effect. So you see, the same neuron, exactly the same neuron, which fire both. So when the monkey sees somebody grasping, have a copy of the same action that she is doing. So then we will see what could be the explanation of that. But the phenomenon is extremely curious because the same neuron which have both visual and motor properties, but specific, not only they just respond unspecifically to visual and to action, but to this, I hope, okay. So some point, before discussing what it means, one point which I want to stress is that it's not conditioning, has nothing to do with conditioning. In these slides that you see here, in A, there is another monkey. This monkey is an intruder, has nothing to do with experiment. She's sitting in her cage and she's eating. Anytime this monkey is eating, the monkey from which we record have a discharge. But of course, the only monkey which got the reward is A, so has nothing to do with reward. It's something which is related to behavior of the monkey. It's been learned maybe during when she was newborn and so on. Well, which is the function of these neurons? When we discovered them, we have a lot of discussion. And the first idea which came to mind to everybody was maybe imitation. If you think about imitation, that's the ideal mechanism. So imitation is something, I will come back later also on imitation, but if you think something, you do something like that, you see, it's a description of this movement in your retina, in your visual system, and yet you can immediately repeat it. So there is a kind of specific kind of link between my action and your capacity to repeat it. There is a professor in Germany, Wolfgang Prinz, who studied very nicely this phenomena and showed how it's precise, this capacity of imitating that we have. Anyhow, there was a problem. The monkey do not imitate. So all the discussion about imitation, when we are talking about monkey, not when we are talking about humans, but when we are talking about monkey, it's wrong because the monkey are unable to imitate with their hand. They are able to make some grimaces, but they don't learn by imitation and they don't use their hands for imitating. So what could be the other possibility? The other possibility is that to have a copy in my brain of your action could be a beautiful system to understand what you are doing without trying to do some complex mentalizing. So imagine that you are in a bar and you see somebody grasping a mug of beer. So what do? You immediately understand what's going on. You don't need to say, oh, there is a head, there is a mug, so maybe this guy is going to grasp. So all this inferential thinking is not necessary. Maybe there is another mechanism. So this mechanism allows you to understand immediately what is going on without linguistic capacity or, in a way, inferential capacities. So how to prove this point? What we decided to do is to prove it by changing all the contingencies around the animal. So if these neurons are for understanding what is going on, if I change all the contingency, and yet the neuron is firing, it means this neuron understands what's going on. Just to make you an example, you are in your hotel, in your room, and you hear some specific noise like this one. You will understand that somebody is walking, no doubt about that. So there are many actions which have specific type of sound. We did the same experiment with the monkey. We presented different noises which the monkey knows, and we recorded single neurons. So here, for example, the experiment that is steering a sheet of paper, and there is a discharge. Not strange because it's a mirror neuron, and the monkey sees and hears the noise. But we can put a screen. So now the monkey doesn't see anything, but hear the sound, and there is still a discharge. So that's our protocol. It's very non-sophisticated data. You just see trial by trial. It's very ugly, but anyway, it's the easiest way to explain it. So you see there is the discharge there. Now, control experiment, white noise, there is nothing. And even noise which are meaning for the monkey. For example, chimpanzee calls, which scare the monkey. They are afraid of chimpanzee, do not produce any discharge. Well, the other experiment, which I am afraid I will be unable to demonstrate in detail, but it's to see if the monkey can understand an action, although the monkey cannot see it. So in this case, you see there is a girl grasping orange, the left panel up, in full vision. and there is a discharge. Then the monkey, the girl, is doing the same thing, but there is no object. So she is not grasping. For the monkey, this has no meaning. The monkey doesn't care about that. There will be no discharge in F5, in the area with. Then we close the screen, and the monkey sees only the beginning of the movement. But the neurons have been selected in such a way, the trigger feature of this neuron is grasping, is moving the fingers. And the discharge is there. I will make only one attempt because well, maybe we're so lucky that I can show you at least one. No, I'm sorry. The presentation of film are not possible, but it's not so important. So anyway, the results show it very clearly that there was a discharge, although the monkey cannot see what the hand is doing. So in other words, the monkey has a mental representation of grasping, although cannot see what happened. So at this point, I think we are on a safe ground in accepting the hypothesis that this discharge is not related to vision or so, but to understanding of action made by others. So here you see some of the people. Luciano Fadiga, Leonardo Fogassi, Vittorio Gallese were my students. They were involved from the very beginning in this experiment. The people on the right did other experiments. They are much younger, but some of them already famous. Buccino became now professor in Catanzaro. Christian Kaiser is professor in Amsterdam. So some of them already have its important scientists. Well, up to now, I have spoken only about one technique, single neural recording, which is the best technique for understanding the mechanism. But if I want to know not only the mechanism, but I want to know also the whole circuit, I have to use another technique. And this other technique is basic, it's called brain imaging. Probably all of you heard about magnetic resonance. Magnetic resonance, like positron emission tomography, are something magic. I think it's a miracle of technology, but the principle and the basis of them are extremely simple. At the beginning of the last century, Sherrington, the father of neurophysiology, found that if you touch the paw of a rabbit and you look what happens in the contralateral somatosensory part of the brain, of the cortex, you see an increase of blood flow. So anytime I touch here, there is more blood flow here. It was well known. Immediately after the Second World War, there was a Russian scientist working in America, Sokolov, who tried to put some radioactive material. And of course, radioactive material, if you put it a certain way, will concentrate where there is more blood. And he could demonstrate that, so using some detector of radioactivity, you could find this increase of blood flow. That was the beginning. After that, there was just miracle of technology. You can localize this, you can use a different way to see if it's in the depth and so on. And then the other miracle was the spin of electron and how to reconstruct brain using the spin of electron. It's really something magic, because when you see, it's exactly the brain as you see when you cut it. And yet it was just done because there is a difference between, for example, the part of the brain which is rich in lipids or rich in water and so on. Anyway, this is technique which allow us to know where is more blood, so more activity. We used recently this technique also in the monkey, and that was somehow a surprise, because people said, oh no, in the monkey it will be impossible because the subject must be very still in the scanner. Otherwise you have artifact due to magnetic field. Well, but the monkey, if you train to fixate a point and you reward when the light changes, for example, it stays for half an hour, 40 minutes, very quiet inside the scanner. So that has been done in Belgium together with Professor Urban and his collaboration. And when the monkey was there, fixating a point, we presented some films, some video clips. For example, a girl grasping something. Very boring film, but anyway, they were effective. And in this way, we were able to found, I don't think you can see, but anyway, if the light was better, and maybe it's not important for you, but certainly you see these red spots on the, especially the lowest row, which has been done with very strong machine at 3 Tesla. You see this red spot. This red spot is where there is activity when the monkey observes somebody grasping an object. And there are essentially 3 spots, maybe this will be better, 3 spots. One is still a visual area, which is called STS, and then there are 2, parietal lobe and frontal lobe. So this is the circuit which is able to organize your action and transform it into my action, step by step. First, your action is coded visually in STS, and then it starts to be coded in motorical term in parietal and frontal lobe. Well, now I think the time is running, and I will shift to human. As soon as we discovered it. We discovered it in '92, and then for 2-3 years we did control experiments because we were very worried that it could be a sort of artifact, that the monkey was doing something strange that we were unable to understand. When we were convinced that it was true, we tried to see if the same thing is present in humans. So if humans have a mirror mechanism. So the first experiment has been done in Milano, in San Raffaele, together with Professor Fazio and Daniela Perani. So we did the first experiment, and it was really a very dramatic experiment. So at the time, we used radioactive water, and the life was very short. So there was one of our students running from the cyclotron to the monkey, to the human, to the students, injecting it. And sometimes the amount was very little, we cannot see anything. But anyway, the results were quite good. And now I will show you a meta-analysis, which has been done again in Germany recently. It's something like 100 studies, which show exactly what you saw in the monkey, that the human mirror system is present in these 3 areas, essentially, and for grasping, for hand action. And I am very happy about that, because some psychologists at a certain point said, well, but the human is different, this system certainly is very important, but not for human. That's poor Darwin, it's exactly Darwinism. So the point which are present in the monkey remain in humans, because that's important. I mean, when nature discovers something important, it doesn't change. It remains. And this mirror mechanism has been discovered probably very early in evolution, but remained in the monkey and humans practically in exactly the same spots. Well, of course, you have to recognize mouth and hand and food action, and so you have different spots. That's explained why we have such a big areas involved that indicate, for example, if you see here, mouth is more ventrally. If you, again, I have not the pointer, but look at this hemisphere on your right, which is the left hemisphere. The mouth is very low, then hand is higher, and the foot is even more higher. If somebody has some kind of neurological training, we remember that's exactly how the motor system is organized. So there is this homunculi with the foot up and the mouth down. Well, recently, again, with this group in Leuven, which we work together, we tried to see if there is a specific region for tools. So we look at, in monkey and humans, where are the areas for tool recognition? And when we observe somebody using a tool, in humans there are 2 activations. One is a kind of generalization of what you're doing. So it's activation of the same system which is for grasping with the hand, because the goal is the same, and so it's the same. However, we humans, but not the monkey has nothing of this kind, have also a specific area which is located in the inferior parietal lobule. Maybe you see with different tools. And that's interesting for neurologists, because the lesion of this area produces a specific deficit, which is called idiomotor apraxia. And that's the part where coded our capacity. It's also interesting that although you teach a monkey to use a tool, monkey from evolutionary point of view is enabled to construct tools. And this area is present in human, but it's absent in monkey. Again, the Darwin notion that there is evolution also, there is not only repetition of something. Well, now I want to tell you, just for break, for a moment this neurophysiological data. There's a story about a psychologist from Oxford, very good psychologist, who at a certain point had a sentimental crisis. So he decided to go away from Oxford, and he went in Africa to Rwanda. In Rwanda, there is a big colony of gorilla there, and decided to study gorilla, the behavior of gorilla. Gorilla have a very big brain, and he looked at the gorillas and was very surprised that they don't do anything intelligent. So the gorilla wake up in the morning, they eat, they walk, they do something, but they have no enemies, they are big beasts, they have food, plenty of food, so they do anything intelligent. And Nick Humphrey, who is the guy I mentioned before, said, there was no obvious sign of the gorillas using their intelligence to any practical advantage. Hard as they looked, I never saw them do anything that struck me as being clever, let alone any sign they were having to solve difficult conceptual problems. Now, if you think to people, general laymen, why is brain for? They would say, oh, well, for solving difficult conceptual problems, or anyway, for something complicated. So why gorilla, who has a very big brain, don't solve anything? Why they have a brain? The brain is a costly structure, a lot of metabolism. So what is the answer, according to you? So he said, I wish I knew why my brain is for. So why do you think the gorilla have such a big brain when they don't do anything apparently intelligent? The answer is very simple. Social life. It's true they don't do anything intelligent in terms of conceptual problem. It's true they have no enemy and food is plenty, but they have a very complex social life. They make alliance, they change alliance, they make war for women, for food, for anything. So the life is very complicated from a social point of view. But that's why this part of brain evolved, because social life is very important. An ethologist from St. Andrews said that the gorilla brain is Machiavellian brain, because what is important is to read the intention of others, to prevent them, and to understand what's going on. So that's why we have such a big, we have the same thing, we are very interested in the fortune of others, as was said by Adam Smith. Okay, now I want to clarify something which I don't want to lead you in the wrong way. So I'm not a guy who is dogmatic. So I think we discovered one mechanism which is very important in understanding the other. But that's not the only way. There are some occasions in which there is no doubt we have to use other type of mental possibility. Now, this experiment, which I will show you now, demonstrates that we can understand animals in this case, but this is true also for ourselves, using different mechanisms. So in this experiment, which is an fMRI experiment, a brain imaging experiment, there were two parts. In the first part, the students was looking at somebody of three different species. So either a student biting an apple, or the monkey doing the same thing, or the dog doing the same thing. So the action was the same, biting. That's common to all of three of them, of three species. The other part of the experiment concerned communicative gestures, communicative actions. So the students that you see here was reading a newspaper. There was no sound. There was only the lip reading. So the student in the scanner has to try to understand what happened. Or the monkey was doing some particular communicative gesture. It's called lip smacking. It's considered an affiliative gesture. And the dog was barking. No sound, just the movement. Well, the results are here. So you see for biting, I don't know if you can see well, but if you look at the left hemisphere, it's exactly the same. So although the phases are different, the left hemisphere indicates the action was coded in the same way. So my biting, the biting of the dog, and the biting of the monkey are coded in the same way. I understand biting because I know what means biting. But now, let's look at communicative gesture, which are species-specific. So we don't bark. And so you see, humans understand when somebody else is reading a newspaper, using specific part of the brain, Broca's area and so on. For lip smacking, there was a small activity of the same area, then I will explain you why we think it happened. But if you look at the left hemisphere of the dog, and even the right hemisphere, it's white. So there is nothing. So only the visual system was active. What it means? That we understand barking not because we are using mirror system, but because we have a visual representation of the action, and then we think about it. There is no way in which, I can say pow, I don't know, in any language, pow is different, barking is different, but there is nothing inside myself which corresponds to barking. I don't bark. So in this case, we understand barking not because we have the same motor program, but because we know what it means. It's what the animal is doing. Let's imagine something more strange. Let's imagine an alien person will arrive here, somebody from Mars, and he became from time to time green. After a while, decide to say he became green when he's breathing. But you will never understand by yourself what it means to become green, because you don't have this type of capacity. So we have to be distinguished, not to be dogmatic. We have one way of understanding using this basic mechanism, but then of course for some other type of behavior, we have to use our capacity of inferring science and so on. So what we found in our experiment, something which has been proposed by phenomenologists, by Husserl, by Merleau-Ponty, by Sartre. So here is the sense of gesture is not given, he wrote, Merleau-Ponty, but understood that is recaptured by an act on the spectator part. The communication or comprehension of gesture comes about of the reciprocity of my intention and the gesture of others. It is as if the other person's intention inhabited my body and mine his. So that's the point of phenomenology. It is absolutely correct. That's the basic way in which we socialize. But then there is the other way that I mentioned before. Well, one point which I just said at the beginning, I said the monkey do not imitate. So the mirror mechanism cannot be as evolutionary basis of its imitation. However, we discovered something strange, which at the beginning we have not understood. We tried to show the existence of mirror neurons in humans using another technique, which is called TMS, or transcranial magnetic stimulation. So if you apply a magnetic field to your motor cortex, you can elicit movement on the other side of the body. So left stimulation, right movements. Well, if you decrease the threshold, you don't see anything, because the threshold is too low. Now, if you ask a person in front of you to grasp an object, you see that now the threshold increases. And the same threshold that before was inefficient, now it's enough to produce movement. So we demonstrated that there is an increase of activity when you see another. The experiment was very elegant. It has been done by Luciano Fadiga, essentially, and Pavia, a neurologist in Parma, because it was exactly the same muscle which are important in order to grasp, which are used in order to grasp, that became active. So we have a kind of muscle-to-muscle transformation. I said that we have not understood fully it, because when we did this experiment, we were very happy that there was. But if you look, there is one condition, the third one, in which the person was not doing grasping, was not doing an object-directed action, but was simply moving the arm. For the monkey, that doesn't mean anything, but apparently for human, yes. But so it was a jump from what is called in neurology transitive gesture. I grasp something, I push something, to intransitive gesture, which then we use as symbolic, victory, okay, and so on. Or we simply use as mimicking, I open the door. So we have another type of mirror mechanism, which transform not something which already dedicated, like grasping, but which is undedicated, so just a simple movement. Why that's important? At the beginning, as I told you, we have not understood. Because it's basic for imitation. If I ask you, for example, do that, can you do that? Please, up. No, just what I do, up. Okay, now I do in the opposite direction, but you do up. It's easier or more difficult? Much more difficult. So we have the capacity to transform this intransitive gesture immediately in our own. So what is the mechanism? It's still the mirror mechanism, but not the one which we discover in the monkey, but a specific one which is present in human. Recently, well, here is an experiment that has been made by Pasquale Leone in Harvard, which showed that our capacity to follow the movement done by another person is extremely precise. So the activation of our motor cortex follow exactly the movement of the other. But anyway, that's a very important experiment for experts, to say. So that's a book which has been published recently by Ramachandran. Ramachandran, maybe you heard, because he wrote also a popular book, very nice. There is one beautiful about so-called phantom limbs. So Ramachandran is an Indian who graduated in Oxford and now is professor in San Diego, in California. And he wrote, culture consists of massive collection of complex skills and knowledge which are transferred from person to person through two core mediums, language and imitation. We would be nothing without our savant-like ability to imitate others. And this chapter, it's called The Neurons That Shaped Civilization. So his idea, and I fully agree, is that the mirror mechanism was on the basis of our capacity to imitate, and therefore of our civilization. Because what means civilization? What means culture? Culture means that something that you discover and the next generation is able to do it. Why animals have no culture? Because they are unable to reproduce. So if I am able to reproduce something which my father, my grandfather, and so on did, the culture will be not here. Our culture is based on imitation. And that's something one has to tell teachers, because at least in Italy, after the 68, 70, it was said that imitation is bad. One should create, one should be creative. It's not true. One has to be, first of all, able to repeat what the other people are doing. If you think about Leonardo staying years with Verrocchio, if I remember well, repeating the same thing. Recently, I have visited by a Belgian artist, Jan Fabre, who said that in Royal Academy, they do for, I don't know for how months, they replicate Dürer, because that's very difficult to replicate Dürer because of his precision. So imitation is something fundamental. Now, emotions. That's something which really, it's again back to Adam Smith. He said also we are not only interested in the fortune of the other, but also happy when the other are happy, and unhappy when the other are happy. Is this true? Well, yes. With emotion, we did the following experiment. Again, and brain-imaging experiment. We presented different odorants to students and to see what is the effect on them. And then in the second part of the experiment, we presented faces, which would express the emotion relative to different odorants. I must say at the beginning, the experiment was not very successful as far as the positive emotions are concerned, but was very successful with negative. And one emotion is really extremely strong, repeatable, and we use it very much. It's disgust. Everybody who has this odor, feel the odor of rotten eggs, make this grimace. It's something which, that's true for female, for male, for old people, young people. It's universal. And if you think from evolutionary point of view, it makes sense. Because if somebody is disgusted, it means that this food cannot be eaten. It's dangerous for you. So from evolutionary point of view, disgust is very important. Well, we did the experiment with disgust, with the fMRI, in two parts. First, odorants were given to students. Second part, it was presented faces of somebody disgusted. So we have a part of the brain which is specifically involved in emotion and which is controlled by this structure which is called insula. And now, well, again, I don't know if you see, but anyway, you see this red spot. This red spot, it's for unpleasant stimuli. They are both on the right and left hemisphere. The crucial thing, however, is here. Here we presented faces of disgusted people. And the white spot are precisely the voxels which were active both when you feel a bad odor
and when you see somebody presenting disgust. So we have exactly the same neural structure which is active when I am disgusted because for physical reason I am disgusted, or when I see you disgusted. So I feel with you. It's not another part of the brain, and then I say, oh yes, I understood that he is unhappy, so I am also unhappy. But you are really disgusted because it's exactly the same brain part. 1 year after us, approximately, in London, it has been done an experiment with pain. Tanya Singer is the author, the senior author, and she discovered exactly the same thing. So if you feel pain, or you see the pain in your partner, they use the girls and the partner. The girls feel the pain, and then they look at their partner and be shocked. Well, it was the activation, again, of the same area, insula, and another structure, which is called symbole. So we published this paper saying, we are both disgusted in my insula. Again, one should not be dogmatic. Of course, I can recognize emotion also using other means. When somebody sends me a message and there is an emoticon with a face which shows pleasure, I don't feel anything. It's something conceptual, symbolic, I understand what it means. But we have also mechanisms which really, so when I see somebody feeling pain or in pain or expressing other type of emotion, I have inside myself the same emotion. So we are really linked emotionally one to another. Then culture can modify it, can do many things, but we can discuss about this later. Well, I want to finish with something which we did recently, a couple of years ago. Maybe you are aware there is a disease called autism. The core symptom in disease is deficit in communication, in the capacity to socialize with the other people, and then there are other symptoms, like they have, for example, moments which become very angry, or they don't want to be touched, and so on. But the core symptoms is this capacity, this incapacity to socialize, to be with the other. To give you an example, some of them are very intelligent. They may have very high IQ, but still they cannot understand something very trivial. Can you pass me the water? They will say yes, but they don't understand what it means, because literally they can't do it. But they don't understand that socially it doesn't mean if you can, but give me, please. So that's the type of disturbance, even in those extremely intelligent. Some have IQ which is low, some are very bad IQ. So we used people which have normal IQ, and we did the following experiment. It has been inspired by an experiment in the monkey which I have not mentioned today. But anyway, here's a boy here who has to grasp an object and to put either in his mouth, it's a piece of chocolate, for example, or to put into a container. It's a piece of paper. And we recorded the activity from the muscle which opened the mouth, the muscle myloideus. It's one of the muscles which opened the mouth. And we look at how the motor system is organized in a normal, typically developing kid. So a typically developing child points, you see this red curve and blue curve. The red curve is the image of myloideus when you grasp in order to eat. The blue when you grasp in order to place into a container. So look at the red lines. Point 0, it's the moment in which the hand touched the piece of chocolate. So you see that almost 1 second before, this muscle is already excited. Why? Because when you want to grasp a piece of chocolate, you know what you want to do. So your intention is clear for yourself much before than you. So I want to grasp, I move, then I touch. And this is reflected in truths in your motor system. So it's already 1 second before, approximately 100 milliseconds probably. And that's what happens, still in typically developing children, when they see somebody else eating chocolate or pudding. So again, when you see, when these normal people see somebody eating a chocolate, there is an activity inside their motor system. So the action of others intrudes in the motor system, and there is this increase in activity, which is very early, and then it disappears because it doesn't matter. So that's the pattern in typically developing kids, so kids without neurological disturbances. And here in autistic children, the first thing, which I think is very clear, that their own intention is not clear for them. So logically, they want to eat chocolate because they grasp it. They are intelligent. Some of them are very good in school, as a matter of fact. But their capacity to transform their intention into motor activity, it's not normal. Only when they have the chocolate in their hand, they want to put it inside the mouth. So instead, it's like robots, you know, the robot of all generations that move by step. But the most important thing is here. Here they observe other kids eating. There is nothing. If you ask them what they are doing, they are grasping chocolate, they are grasping a piece of paper, they are eating it. So logically, inferentially, they know everything. But inside their motor system, there is no empathy. So their empathy is zero. They have no connection with the other people. So they are living in a world which is not made of people, but of object, of action, and so on. But the activity per se of others is ineffective. Here is a sentence from a lady who is now in his 40s. She's autistic and she's intelligent. She wrote her life when she was young, she's autobiography, and she writes, I found it hard work to move. He has a father which was continuously saying, do it, why you are so slow, please do it. So I was trying to really to force her. And so I found it hard work to move at all, as I had to think out everything I did in order to be able to do it. In a way, I had to order my body to carry out what it had to do by thinking it out all the time. So you see their capacity to transform their intention in action was impaired. And that's one of the basic deficits. But if you have that, you have a problem also with the others. If you don't know how to transform your own intention in action, even less you have the capacity to understand what the other people are doing. Again, one has to be very careful. My friend in California published several papers, both Jacoboni and Ramachandran, saying autism means broken mirror neurons, which for a doctor is really, no, it's much more complicated. It's a genetic disease with a lot of motor system problems. So one, however, point is that these people, even when they are highly intelligent, have still an enormous difficulty to understand the other from their, in the natural way. So they understand the other if they think about them. Recently, with Daniel Stern, who is a famous psychologist and psychiatrist, we look at the capacity of children to recognize what he calls the vitality form. Vitality forms are not real emotion, but this small gesture that we exchange all the time. If you have a couple of fiancés, you will see that they are kind one to the other. They give the newspaper in a certain way, and vice versa. If a couple which has bad relation, you will see that they will interact in a particular way, which for us will be very obvious that something is wrong with them. So for normal children, it's very easy to understand if you present films in which two people have a good relation, they like each other, how they give object, how they touch. For people with autism, they do a lot of mistakes. So for them, for the group we have, was no difficulty to say it's touching, it's grasping, and so on, but to understand what is the relation between the two. It was 25–30% of error in tasking, which is extremely simple for normal kids. And that's another aspect of autism, this incapacity to understand the feeling of the others. Well, I think I gave you a panorama, a view of what the mirror neurons are doing. So they allow you to be in contact with the other, both in what psychologists call cold actions, like grasping, and so imitation, and so on, but also in the emotional aspects of our life. Okay, thank you.